I left you somewhere around here last week talking about the structure of individual nerve cells and their function. And I told you that the individual nerve cells are basically analog to digital devices, so that there's signals that they send out to their target. They send their signals out through a cellular process called an axon to axon terminals. And this could be, the target of this could be another neuron, or it could be a muscle cell or a gland cell. Okay? And that the message is encoded digitally. Okay? So there is literally a wave of activity that spreads down the length of the axon. And that wave is the opening and closing of ion channels. So ions move across the membrane. So you can think of this, and sometimes it's called an electrical signal, but it's actually the flow of charged ions that occurs. And the message is encoded. Remember, I mentioned that the message can be the content of your dreams or your memories or signals sent to muscles, all different kinds of things, but it's all encoded in this digital message of ion flow. The components of that message are called action potentials. And that word potential refers to the change in charge across the axonal membrane. So you have these individual action potentials happening. And when you do research on this kind of activity, you can hook up these changes, these action potentials. You can literally listen to them because they're charge or voltage changes, and you can hook them up. You can hook up your experimental apparatus to a speaker and drive a speaker just like you would in a stereo system, and you can hear the activity happen. And as these action potentials occur, you hear a pop sound, like that, or depending on the frequency of the message. Okay? So I guess another way to say it is that all of your dreams and your memories and your activities with regards to muscle activity are encoded in pops and like that. Right. I also mentioned to you that the axons, many of the axons are covered by an insulation, just like an electrical wire. So that in an electrical wire, you don't want it to short out, and so you insulate the current flow. And the current, in the case of electrical wires, is the flow of electrons. In this case, it's the flow of ions. But the similarity is that something that's charged is moving. Okay? And so you have insulation that is present around these axonal processes. And this is called myelin. There are specific diseases that occur, like multiple sclerosis, where you have a problem with your myelin, which means that you have a problem with the insulation of your neurons. And they don't function as well. So they don't send their signals. In the case of multiple sclerosis or muscular dystrophy, they don't send their signals effectively to the target muscles, and so you have problems in locomotion and other things. So you've got an insulation around the axon, and you might notice that unlike an electrical wire, this insulation is not continuous. So there's gaps. There's insulation and then a gap, and then insulation and then a gap. And what happens at these gaps is that the action potentials are regenerated at each gap. And so you could think of this activity in the axon is jumping from gap to gap to gap, and it moves very quickly. In the fastest neurons, the message goes at about 120 meters per second. About 120 meters per second. Other neurons are a little bit slower, but that's the fastest you can do, and that works, right? Because you can dream, and you can remember, and you can locomote effectively, right? This is a picture of a cross-section of a neuron, a neuronal axon. So here's the axon right here, and here's the myelin around it, and you can see that it's in layers. So there's these layers of insulation around the axon that prevent it from shorting out, so to speak. And then the action potentials are regenerated from node to node to node. And in the language of neuroscience, they call this a saltation or a jump. From the Latin saltare, to jump. So it jumps down the axon at about 120 meters per second. What's shown in this is a very simple pathway that you might have in a reflex. And I think yes, I did. I sat up here last week and reminded you of the knee-jerk reflex so that the physician smacks your patellar tendon with a hammer and you kick. That's a very simple reflex that involves just a few neurons. And what's shown up here is just such a pathway. You have a sensory neuron right here, and this would be the one that's, say, out in the muscle and senses the stretch of the muscle tendon. And then that sends signals jumping down the axon back into the central nervous system. And in this case, the portion of the central nervous system is the spinal cord. So that message is sent from my patellar tendon into my spinal cord. And then there may be a few neurons in between, and then the message is sent out by a motor neuron out to the muscles in like my quads, and so I kick. Okay? So here you've got a pathway that has 1, 2, 3, 4 neurons in it. It's got a sensory neuron, a couple of interneurons in between, and then a motor neuron that goes out to the muscle. There are the response that I elicited by throwing confetti into the audience last week involves thousands or tens of thousands of neurons. But that information is processed pretty quickly. Okay? There was a little bit of squealing, a little bit of screaming, and then some laughing. Right? So that information can be processed pretty quickly. This is to show you that there are multi-neuronal pathways. So one neuron sends its axon to another neuron, and then this neuron sends its axon to another neuron, and this one to another. Okay? So like in the spinal cord reflex there, you have a sensory neuron connecting to an interneuron, and then another interneuron, and then a motor neuron. In the case of the confetti, you had sensory neurons from the eye connecting back into the brain, and then lots of interneurons, and then some connections to skeletal muscles so that people would kind of move away from the confetti, or they drive their respiratory centers to laugh, that kind of thing. Now, I also mentioned to you that there's this electrical or ionic activity that happens in the neuron, and then that is changed into a chemical message at the target. Whether that target's another neuron or whether it's a muscle cell or a gland cell, the flow of ions is changed into the release of a chemical, and that's called a neurotransmitter. Okay? So here you have the very end of an axon, and here's the action potential coming down, and then you have these little bags here that are called synaptic vesicles, and they contain the neurotransmitter, the chemical, right? And you have these little vesicles that will combine or fuse with the membrane here and then release the neurotransmitter into this space. So there is a physical space between one neuron and its target. And you have the release of the chemical, and then it diffuses across the space, and it binds to receptor molecules on the target, whether that's another neuron or a muscle cell or a gland cell. As you might expect, if you think about this a little bit, This right here, this junction is the subject of a huge amount of research. How do neurons talk to their target? The pharmaceutical companies are really interested in this because if you can control the message as it's being sent from one cell to another, you can do things like control pain, or you can restore function between a neuron and a target, if you can manipulate this connection. And the junction here is called a synapse. So this space is called the synaptic cleft. Right? So you have a flow of ions down the length of the cell into the axon terminal, and then the release of a neurotransmitter that takes the message to the target to contract a muscle cell, or to turn on another nerve cell, that kind of thing. Or to turn off a nerve cell, somehow to affect the target. Okay. All right, now, it takes years to develop the basic plan, the basic structure of the nervous system. When you're born, you're definitely not fully functional. Okay? You're partially functional. There are a lot of things that are working, but they're not working to optimum. So there are milestones along the length of life, from embryonic life all the way to death. There are milestones that occur in the development and changes in the nervous system. Most of you, this shouldn't be surprising, shouldn't be, but most of you are not fully functional yet. Right? And I was fully functional some time ago, and now I'm aging, right? So I'm not as functional as I used to be, but I do okay. This is a quote from a very famous neuroscientist around 1900. And this gets me back to the synapse, the junction between the neuron and its target. This is Santiago Ramon y Cajal, and he was a Spaniard. And one of his contemporaries was an Italian, Camillo Golgi. And they had arguments, big time arguments, over the years about whether or not there was a synapse. Neither one of them had seen a synapse, but they knew a lot about the functioning of the nervous system. And Santiago Ramon y Cajal said there is a gap between the neuron and its target. And Camillo Golgi said, no, there isn't. And here you have, if I may do this, here you have a Spaniard and an Italian. Woo! Letters back and forth, accusing each other of all kinds of things. They both won the Nobel Prize. They both won the Nobel Prize. The Spaniard won, okay, in the sense that he was right. There is a junction, but there was no proof of that until the 1950s, and both of these men were long dead. So they had their arguments based on some data, but no one had ever seen a synapse until the 1950s. Whoa, Santiago Ramon y Cajal was right. This quote is one of my favorite quotes in science. And it's sexist, okay, that's 100 years ago, right? Every man can, if he so desires, become the sculptor of his own brain, or her brain. As I speak, right now, I am changing the structure of your brain. I'm changing you. Sometimes students go, it's okay, I'm a trained neurobiologist. Whenever you communicate with someone, you physically change the structure of their brain because you change the structure of their synapses. You can do this in a good way, you can do this in a bad way. The point is that you actually change the structure of the brain. You do this, you change your own structure. You're constantly changing the structure of your brain. You're strengthening synapses, you're weakening synapses. For instance, you think, why would you want to weaken synapses? Why would you want to disconnect? Oh, there are lots of reasons. Okay? If you remembered everything that happened to you after you woke up this morning, right away, constantly, you'd go insane. Way too much information. Way too much information. So you get the information, and then you utilize it, and then you disconnect. Hopefully you don't disconnect everything. You need to keep connections, and in some cases strengthen them. Like maybe a little bit, understanding the structure and the function of the nervous system. Strengthen those a little bit. When you learn a motor skill, when you learn how to ski, you're strengthening connections. You're making new connections. And what was this, maybe about 10 years ago, something like that, scientists actually have videos of shape changes in neurons. They move a little bit, just a little bit. So they may retract an axon or extend one a little bit. So your nervous system is changing constantly, and you're allowing it or not allowing it. You're weakening connections or strengthening connections. It almost sounds exhausting, but you do this all the time. It's what you do when you learn. It's what you do when you dream. It's what you do when you think. It's what you do when you locomote. So you actually can, you guys are all sculptors. You're making your brain right now. Hopefully you'll do a good job. You will. So here's a picture of development in the central nervous system and spinal cord. The central nervous system, which is brain and spinal cord. And it starts out as a long tube. So you actually, right in the middle of your spinal cord and your brain, you have a fluid-filled space. You may have heard of cerebrospinal fluid, or CSF. CSF is a diagnostic tool. Physicians can monitor any changes, mood changes, and those may represent disease states or whatever. So it's something that physicians pay a lot of attention to. But you have a tube, and then the tube is lined by tissue on either side, or all around it, actually. And then over time, 3 weeks, 4 weeks, 7 weeks, 3 months, 6 months, newborn, the tissue cells grow and they divide. More cells, more cells, more cells, more connections. The cells actually move and migrate within the developing tissue. Okay? You develop particular regions, like a forebrain and a midbrain and a hindbrain. The forebrain, very generally speaking, has to do with cognition and thinking and visual and auditory reception, those kinds of things. The midbrain and the hindbrain are generally more reflex areas. In your hindbrain, you have particular sets of neurons that drive your heart or drive your respiration. So the basic functions, generally speaking, are down here, and the more advanced or complicated functions are in the forebrain. So not only are there physical changes in the brain, but there are functional centers set up as the brain develops. And then once you're born, it still keeps changing. Still keeps changing. So during fetal development, you have 9 months in the womb, right? The trimester is 3 months. So you have a first trimester, a second, and a third. And this gives you some very basic functions that occur. In the first 3 months, the connections begin to be made between the neurons, between the millions of neurons. They're dividing, they're moving, they're connecting. Right? In the second 3 months, the connections are made in such a way that they're targeting the muscle, and you get muscle movements within the womb, within the uterus. And in the third trimester, sensory pathways are forming. So visual pathways and auditory pathways. Pathways with regards to olfaction, smell, or touch. All of these are beginning to form at that point. It's very interesting to think about the last 3 months in the uterus as a time in which you begin to sense things. It continues once you're born. Visual and other systems start to mature. Sometimes in a developing nervous system, a lot of contacts are made initially, lots. And then, interestingly, some of those are removed. Okay? So it's like you make all of these, lots of connections, and then some of them are removed as you develop. Pathways become more specific. Along with this maturation is myelination. So at first, the pathways are not insulated, and they can work, but the messages are not sent very quickly. The messages can be sent, but not very quickly. When myelin begins to develop, then you get the insulation and you get up to 120 meters per second. So now the signals can be sent faster and faster and faster, and you can respond more quickly to sensory input. Either that response could be forming a memory, forming an internal image. It can also include responding in a locomotor fashion. So pathways become more functional. 100 billion nerve cells. Remember, last time I talked about how there was a book that was written called The 3-Pound Universe. You contain the universe within your brain. You contain information about the universe. You contain information about the edge of the universe, or maybe that there is no edge to the universe. You contain information about the structure of atoms, the internal structure of atoms. You contain all of this information based on the activity of these nerve cells and their connections, strengthening them, weakening them, depending. So, if you think about an infant and a child, they begin to develop skill sets, motor skills, language skills, emotional skills, the ability to remember things, okay? A sense of self, me as opposed to the rest of the world. You begin to explore the world with your appendages, with your hands and your feet, with your hands and with your feet, bringing in more and more information. A lot of things go in the mouth. Okay? As a child, you begin to explore the world with your tongue and your mouth. There's a lot of innervation to the oral cavity, and those pathways are set up and myelinated very early, as opposed to the pathways that drive your appendages. Those come more slowly. But this is a very important and early sensory surface, and so you use it. Like putting blocks in there, or turtles, or whatever you do as an infant. You explore your environment. And then the complicated biology of an internal self, of me versus the rest of the world, versus longer stronger. Me in relation to the rest of the world. No one knows exactly how that occurs, but it's all in the function of neurons. And some might say this is a very mechanistic way to look at life. Action potentials and nerve cells, mechanism. Life has to be more than just mechanism. Just mechanism? That's the wrong word. Life is mechanism. It's an amazing mechanism. Amazing. Just mechanism. How do you do these things? Who knows completely? Know a lot, but not completely. Synapses, as I told you, are sculpted or strengthened. It's sculpted in the words of Ramani Kahal, or strengthened by experience. And I should put up there, sculpted, definitely sculpted. They can be strengthened, they can be weakened, depending on what the point is. Teen years. If you think back to high school a little bit, and you guys should be able to do this easily. I can't do it as easily, but I can do it. This myelination and growth of the nervous system is happening very quickly at this point. It's going to continue into adult life, but it's happening very quickly, right? The brain is still developing. Very generally speaking, 10 minutes, I got you. Very generally speaking, if you look at someone at sophomore level or so, generally speaking, their locomotor skills are definitely not as good, generally speaking, as when you're in your senior year. Right in that time frame, there's a lot of myelination going on. And so you become more coordinated. The term sophomore is kind of derogatory. Uncoordinated, ungainly, not very sophisticated, sophomoric. You can use that as an adjective. But by the time you get to senior status, you're more myelinated. You can think whack, whack, whack. You can think quicker. You can act more quickly and in a more coordinated fashion. But there's still a lot going on. Myelination continues. All of these systems are still ramping up. Okay? By the time you get into college, it's not completed, but things, for instance, like your ability to maintain attention is generally speaking stronger than it is when you were in high school. Just because your brain has matured. It's different to a certain degree for different people, but still you see that trend. During this time, there are disease conditions that appear because the brain is not developing correctly. Schizophrenia appears early on, late teens and early 20s. Can appear, doesn't necessarily, right? Most people aren't schizophrenic. Depression, addiction, all of these kinds of things can begin to occur at this time, depending upon nature and nurture, okay? Depending upon brain development, and also depending upon the situations in which you find yourself, the social situations in which you find yourself. So it's an interaction of both of these. Oops. Actually, late teens, early 20s, apex, as far as brain function, generally. You begin to lose it a little bit. Women keep it longer, generally. Memory systems start to decline a bit. It's a complicated situation. You still have new synapses in language and perception centers. It becomes a little harder to learn languages as you grow older. A little harder. Do it. It's a little harder. Look at that. Myelination continues up into your 30s. Then you begin to, over this, after your early 20s or so, you begin to lose some of the cells, you begin to lose some of the synapses. One of the things that's really, really important is to remain physically active. Okay? What that means is maintaining or increasing sensory input by being physically active, by being mentally active, by being active. So you want to be an active learner throughout your life. You want to be a physically active person as much as you can during your entire life. So you're constantly stimulating the nervous system. And even though, generally speaking, the systems are beginning to fail, you can still acquire new skills and learn new things. And being mentally and physically active is absolutely essential in that. Do you have 5 minutes? 5 minutes. Plasticity is present throughout life, which basically your brain is plastic, which means you can remove connections and make connections. You can change your brain throughout your entire life. Even though, generally speaking, the systems are beginning to fail, you can help maintain those systems, at least to a certain degree, by being mentally and physically active. Later in life, and these kinds of things are being seen more often now because our lifespan is increasing. If you look back into in the United States, if you look back into the 50s and 60s, someone who was in their 70s was generally considered old. 80s was old. Okay? Now, you see a lot of people in their 80s. You see a lot of people in their 90s still mentally and physically active. So as we extend the length of our lives, we also encounter disease conditions that will appear if you just live long enough to let them happen. So Parkinson's, which has to do with the muscular system, and Alzheimer's, that has to do with memory and cognition. These kinds of things appear late in life, and there's a genetic propensity. You can carry the genes through your family where these will appear or not. So physicians now look very carefully at genetic history to be able to predict with some degree of confidence what kinds of challenges you might have as you age. I'm hoping that I'm presenting a hopeful view of life. I mean, there are these challenges that you encounter, but you can prepare yourself well to meet those challenges. And one of the things for college students, I think, is that you're in classes, you're spending a lot of time studying, all good. But I think you have to remember to stay physically active as well. And that means physical activity also means eating well. And I know the challenges for me as well. This semester I'm teaching a 7:30 class in the morning and I keep missing my breakfast. That's not good. So you need to eat well, you need to be physically active, and certainly in classes, hopefully, mentally active. All right? All right.